So Death Valley was not a dry place like it is today. It was a big, huge lake. And so in the bottom there's a big lake. And fossils in that area show that there was basically one species of so-called pupfish. This is Cyprinodon, is the name of the genus. But after the last glacial maximum, which was about 24,000 years ago, the glaciers started to disappear. And by 17,000 years ago, it was getting drier. And most of this lake went underground or it evaporated. Now, there's still ancient water, groundwater underneath Death Valley, which is the remnants of this lake. But so there's throughout Death Valley, there are these springs. So where there's water coming to the surface, and there's just only a tiny little pool there. It's not big, but each of these pools around Death Valley Springs, there's several of them, about 10. I don't remember the number, but there's quite a few of them. They have their own species of pupfish, and they're different species. Okay, and so this is a case where the drying up of a lake created a barrier for the fish, and they diverged, and today they are separate species. Likewise, the Grand Canyon. Okay, so there's antelope squirrels on the north side and the south side of the Grand Canyon, but they're separated by this canyon, and they cannot cross each side, and they are each other's closest relative. All right, but they're different species. And so the Grand Canyon split an ancient population of these squirrels. Some of them are stranded to the north side of the canyon, some of them stranded to the south side, and there is no gene flow across the canyon. And they have diverged genetically to the point that they can no longer mate together, even when you bring them into contact. There are reproductive barriers that prevent that. Okay, another example of, I mentioned this before, the Isthmus of Panama. I said, I think the date when it arose is a little bit controversial. It's somewhere between 5 and 8 million years ago. But nonetheless, the Atlantic and the Caribbean used to be connected. There's a free flow of genes across there. But once this Isthmus of Panama has connected North America with South America, there's it's a barrier. And here I showed this rainbow wrasse and blue-headed wrasse. They're on each side of the Isthmus of Panama, and they appear to be each other's closest relatives, but they are separate species. And so the land bridge there is a barrier that arose allopatrically, and we have speciated. This is not the only example on each side of Panama. There's many different marine animals that their closest living relatives are on each side of this land bridge. And so that separated them. They once were the same species, they have this barrier, and they've diverged genetically, okay? All right, now another thing that's sort of related to allopatric speciation is a phenomenon that's called a ring species. And I know of several different examples of this, but we'll talk about one, or I'll talk about two of them. And so this ring species is where you have a population that sort of spreads around a particular barrier and meet on the other side of the barrier, and the two arms that on the other side end up being reproductively isolated, although we can get gene flow between adjacent populations. And so it's not considered two different species, it's still the same species, but the extreme ends of the range have diverged to the point where they are reproductively isolated. So there is, give an example here, get the picture up there, of Instadina. Okay, this is a species of mud puppy, okay, salamander. And there are several populations here in California. Of course, here in the middle is the Central Valley. And so there's populations along the coast and up into Oregon. It was believed that this species originated up in Oregon and eventually has extended its range south. And as it encountered the Central Valley, well, then the populations, they spread they diverged. One group moved down the coastal mountains to Southern California, whereas the other one is in the foothills of the Sierra Mountains. And so they're in forested land down around Bakersfield or south of Bakersfield. These populations do come back together. And at these extreme parts, they are reproductively isolated with each other. This population over here, let's get a pin so I can draw. Yeah, this population here around the Tehachapi Mountains, just south of Bakersfield, they are reproductively isolated with the population that's over here on the coast. All right, and so they are, but yet adjacent populations can mate with each other all around the foothills of the Sierra and going up the coast. These populations along the coast are not reproductively isolated from each other. So, but these two ends of the population have been separated for a long period of time, and therefore they have diverged. Now, theoretically, you could get gene flow from adjacent populations. It's possible that alleles could be passed all the way up to Northern California and down to this population at the southern part of the Sierras. However, that would take millions of years, and therefore it is inconsequential. And therefore, these ends of the ring species, they are reproductively isolated. Now, if something were to happen that these populations up here were to go extinct, then we would have automatically two separate species, because these guys down in the south are reproductively isolated. Another similar type of story is in Siberia and India, namely the Siberian greenish warbler. Now, I don't have a picture here. I read about this in the journal Science not too long ago. Let me draw a picture here. What's happened with this bird is it originated in the south, and it has Siberia, or not Siberia, but the Tibetan plateau. So I'm just going to draw the plateau right here. So this is real high land, and it's believed that this species of bird, it originated in India, and eventually it has been extending its range over the last several thousand years around around the plateau. And eventually these populations have met in northeast Siberia up here. And where they meet, the two populations are reproductively isolated from each other. And so yet all along here, the adjacent populations, they're not reproductively isolated, and therefore alleles can eventually spread all the way around. So this is another example of a ring species where the Tibet has been a barrier, and the populations that are in the extreme ends of them have diverged genetically so much that they are reproductively isolated. And that's sort of related, although we're not dealing here with different species. Ring species, it's called a ring species. Yes, Emily.
So are they different species, or are they this?
Well, since you can get gene flow all the way around through adjacent populations, they're not regarded as different species. They're regarded as the same. And with the warbler, morphologically, at the extreme ends where they meet up in northeast Siberia, they can be told apart, physically, they're morphologically different. But as you go around to Tibet, you find that from one population to the next, they're very similar to each other. And so this is something that's called a ring species. We would have two automatic separate species if all the populations between the extremes were to somehow disappear. Okay, then you would automatically have two different species. But for now, they're considered one.
So
right?
yeah, so is this a type of allopatric speciation then?
Well, it's related to allopatric speciation in that, in this sense, we have a barrier that has kept some of the populations away from each other. Okay, in the case of the in Satina, that is the Central Valley, and with the wobbler, we're talking about Tibet. But so there was a barrier, and as they've moved around that barrier, they have diverged, and this is exactly what we expect to happen with allopatric speciation. So but it's not really speciation yet. Okay,
Okay.
does that make sense?
Yeah, thank you.
Yeah. Okay, now there are some conditions where allopatric speciation is highly favored. And I'm going to give you a couple of examples where that happens. One is in the case of peripheral isolates. Okay, so what we mean by peripheral isolates, I've already given an example of this once before, although I didn't call it by this name. Get rid of clear out the picture here. Okay, so here is the host range of a species. And as I discussed before, why is a species found here and not out here? Okay, it's because of living conditions. Okay, the right resources that can be utilized by the species happens to be in this range. Now, obviously, as you're heading out, the best place where the conditions are most favorable for the survival and reproduction of this particular species is, of course, toward the middle of the range. And as you get toward the edge, you know, the conditions are becoming less and less favorable. But you don't come to just a strong border where conditions completely stop. There may be little islands of habitat that are further out beyond the periphery, where conditions are favorable for this species to live. And so if we have some individuals, I discussed this with the founder effect, okay? But that was talking about changes in allele frequency, all right? But here we have extreme phenotype or genotypes, and if they can colonize this little island, then they are reproductively, we have a physical barrier between the colonist and the main range, and therefore there's no gene flow. And if we isolate this peripheral population long enough, they will diverge and they will become separate species. So we do have these conditions. We call those peripheral isolates. Wait here. Peripheral isolates, because already they originated from the edge of the range, where conditions were probably pretty rough, and therefore they have extreme genotypes compared to those in the center of the range. And if they get isolated, and usually it's a founder population, there's high genetic drift, and that contributes to very rapid change in their genotypes, which can lead to speciation. All right. Furthermore, some of the selective factors that works on natural selection in those peripheral isolates a lot of times is much different than what is in the main part of the species range, and therefore they can. Then this is another example of allopatric speciation, and eventually these may adapt to the surrounding inhospitable habitats and spread. Another type of condition that can lead to large amount of allopatric speciation or favored is island archipelagos. Okay, does anyone know what an archipelago is? When I say that, what's an archipelago?
Isn't it like kind of like a small island that's not like from far away from other land, but it's like a small landmass like next to like a coastal region?
Okay, it's getting close. It's a chain of islands. Okay, so like Hawaii, it's not just one island. There's several islands there, and the Bahamas. That's an archipelago. So it's not just an isolated island, but it's multiple, yeah, someone here says multiple islands very close together. And so a lot of these islands in the ocean, for example, the Hawaii, that started out as well, they're due to volcanic activity, all right? So when the volcano, it's a hot spot underneath those islands, when first of all the volcano built up high enough to rise out of the sea as an island, it was perfectly sterile. There was nothing living there. And so where did the plant and animal life come from? Well, Hawaii is pretty isolated, but with islands like this, and even we can talk about the Galapagos off of Ecuador. That's also volcanic islands that were built up. They're closer to the mainland. So most of the species that happen to be on the Galapagos, they came from Central and South America, or on the east side of the Andes, west side of the Andes, excuse me. And that's where their closest relatives happen to come from. And it's only those animals that were able to get there, okay? So birds can get there. They have iguanas there, and iguanas, sometimes they will ride currents. In fact, the iguanas in the Galapagos, they seem to be related to those in Mexico. And so, and there's a good reason why they reach there, because down the coast of Mexico, the ocean currents move right out into where the Galapagos happens to be. And so a pregnant iguana that be on a tree that is in the ocean can ride it and ended up there, okay? And it looks like it happened more than once. So what happens with these islands is that they can lead to what's called adaptive radiation. And so we can have rapid speciation, a cluster of new species that all have a common ancestor. So let me get a picture here from your textbooks to explain this. So here we have a new island, an archipelago, and initially nothing is there. But if we have a founder effect, okay, few individuals of the species happen to make it to island A. This is sort of similar to a peripheral isolate as well. Here, the ocean is the barrier, but now these individuals can only interbreed among themselves. There's no gene flow back to the mainland and so forth. And so eventually they will genetically diverge from the mother species that's on the mainland. And now we have species A. And I'm not going to say for Hawaii, this is probably not very likely because that's too isolated. But for the Galapagos, we could have another colonization from the mother colony, from the mainland. However, I'm pointing out something else here. So once we get species A here, let's call that island A. We'll call this species, call it species 1. A few of them may hop over to a nearby island. And now we have them isolated from species 1. Eventually they would diverge into species 2. And we may have from species 2, they will eventually go to species, to island C. And they will diverge, have species 3. And they may jump back to, at a later time, to island A. And they're reproductively isolated from species 1. And now we have no gene flow between this new founder and species 3, and they would develop into species 4, and so forth, okay? And in fact, on the Hawaiian Islands, there are something like 200 species of Drosophila fruit flies, and they all descend from a common ancestor, okay? The genetic studies indicate that they all came from a common ancestor. And so this type of situation can lead to rapid speciation. And that was noticed even by Darwin on the Galapagos Islands when he visited them. He saw that, or at least the Spaniards who were there, told him that they could tell where a giant tortoise came from by the shape, which island they originated from, by the shape of their shells. And so each of those islands have separate species of giant tortoises. And that is repeated also with finches and some other animals as well. So this adaptive radiation, it could happen on island archipelagos, And this is, of course, allopatric speciation, but it could also happen in anything that's sort of island-like, okay? For example, in Nevada, there's mountain ranges. And on the tops of the mountains, they catch enough rain to grow trees up there. And so it's a different habitat on the peak of a mountain, but further down, it's desert. And so there's many types of ground squirrels and even some insects that from one mountain peak to the next mountain peak, they are separate species, supposedly arose by allopatric speciation. Okay, so allopatric speciation is pretty easy to understand. What sometimes is not so easy to understand is something called sympatric speciation. This seems to be pretty common among plants. It's less common among animals, or at least we have a hard time being able to see it. Allopatric means that the species range is separated. Sympatric means speciation in the same range. So there's no physical barrier, but there must be some sort of behavioral barrier that keeps certain parts of the populations from interbreeding with each other. And so this is speciation in the same territory. We also use these words, sympatric and allopatric, to describe species ranges, whether they're living in the same place or living apart. But let's get here to sympatric speciation. This is a reproductive isolation without any sort of physical barrier. And so the speciation takes place within the range. Obviously, though, there has to be some barrier of some sort to separate the gene pool into two or more parts, but it isn't a physical barrier. It might be a behavioral barrier, something having to do with behavior, or a genetic change. As I said, this type of speciation appears to be very common in plants, in that many plants are polyploid. That means that they have more than, that they've hybridized with each other and doubled their sets of chromosomes. Okay, instead of being diploid, they might be tetraploid. Okay, that's real common. And if that happens, if we produce a tetraploid plant when the mother plant species happen to be diploid, then in one generation, we have separate species. Okay. So there's two different ways in which polyploidy can arise in plants. One of them is called autopolyploidy, and this is where chromosome sets have increased within the same species. Okay, give an example here from your book. So let's say we have a species here that's diploid. Okay, it's 2n equals 6, but an error in meiosis leads to a diploid gamete. Okay, so this is massive non-disjunction. And so it produces a pollen grain or something like that. It's a flowering plant that is has a diploid gamete. And if another plant of the same species also has a diploid gamete due to non-disjunction, this would happen more often in those plants that are self-fertilizing. And so these come together, and now we produce a new plant that is tetraploid. It has 12 chromosomes instead of the 6. It has 4 sets of chromosomes, 4 copies of every chromosome. And that is automatically it's automatically a different species. A lot of times these polyploids will have different morphological characters. They look similar to the parents they came from. And furthermore, even though it's reproductively isolated and it grows with one individual, plants can propagate asexually by fragmentation, or their roots. That happens quite a bit. And therefore, we can get new members arising that now have the same number of chromosomes, and meiosis would work normally, and they would be able to sexually reproduce. And so there's a lot of plants. It's believed about 40% of flowering plants in the United States are polyploid, and they rose by autopolyploidy speciation. Another form, okay, so within one generation, they could be reproductively isolated and living in the same territory, same range as the parent plant from which they came. And this is just by doubling the chromosomes. Now, allopolyploidy is where we get hybridization between two different species. Because I said earlier that plants are developmentally plastic, more plastic than animals. So this type of polyploidy in animals is very, very rare. So autopolyploidy is doubling the chromosomes within a species, same species, to produce a new species, which is tetraploid. Allopolyploidy is between different species. Okay, and so an example here from your book illustrates this. We have two species, species A, species B. They happen to have, they're closely related, but they're separate, they're reproductively isolated for the most part. Okay, and with meiosis, normal meiosis, now we have N equals 3, it's haploid, and the other species, species B, still haploid gamete, but somehow these two gametes from different species hybridize, and we get a hybrid. These chromosomes now, although it says here 2n equals 6, they are not exactly, they're not homologous, not completely, okay? And therefore meiosis probably doesn't work. This is probably sterile, but it can spread vegetatively, okay? A lot of plants, okay, just draw a plant here above ground. A lot of times with plants, with their roots, is that they can send up another individual from the root, and eventually the connection between those roots will break, and now we have two separate individuals. And so this is asexual reproduction, and it's cloning, but it's very, very common among plants. Now, at a later time, there might be some other error in mitosis in the germline, okay, in the germline cells, where we get non-disjunction, and now we can double the chromosomes so that we have still diploid. This up here, even though this interspecific hybrid, it's saying 2n, I don't really like that. Really, it's n equals 6. But then we get a mitotic error with massive non-disjunction where we get the chromosomes doubled. Now we have paired chromosomes, and meiosis would work just fine. And now this is now a tetraploid. Over here it's saying 2n, but really it's 4n. Let me get rid of this. Let's go ahead and keep this 2n over here, although those two sets of chromosomes are not homologous. This leads eventually to tetraploid. So this is allopolyploidy. And it happens among plants, okay? Now, there's a good example, a real life example of 3 different species of primrose. This is a flowering plant growing in the Columbian Plateau, okay, in Washington State, Oregon State. There's 3 different species, and one of them appears to be a tetraploid from the other 2, okay, the other 2 species, because 2 of the species in that range have the same number of chromosomes, and then the 3rd species is a tetraploid. And this hypothesis was brought up probably about 20 years ago, and there was a researcher who tried to get these, took the 2 diploid species and tried to get them hybridized in the lab. Eventually he was successful. He was able to in the laboratory, got species 1, species 2 hybridized, producing a tetraploid. And when he grew the tetraploid, it was very much identical to the wild tetraploid that was growing out there. And so that reinforced the idea that the 3rd species arose by allopolyploidy, okay? So this is pretty common in plants, but it's not very common in animals. I'm not going to go to too much detail here on wheat, but our plant that we use for food is gone through allopolyploidy at least 3 different times in its history. Some of the ancestors of wheat plants are still growing in Anatolia, in Turkey. Others seem to have gone extinct, but nonetheless, this is a very complicated picture of how wheat has arisen. And I believe that wheat is, let me see here, is a tetraploid. Yeah, it's tetraploid, and it has the combination of chromosomes from several different grass species that lived in the past, or some of these are still alive today. Okay, now as far as animals are concerned, there's very few examples of sympatric speciation. However, there is one story here of sympatric speciation that is in process. They still haven't separated into separate species, but they appear to be diverging. This has to do with the apple maggot fly, okay, Ragoletes pulmonella. That's what the fly looks like. And originally, there were no apple trees in New England, okay? These were brought by colonists from Europe, and but this fly originally lived on hawthorn, okay? Hawthorn is a plant that's related to apple, and it produces a similar type of fruit. And so these flies, what they do is they go to the fruit, they're attracted by the smell and the taste, and they will lay their eggs on the fruit that it's rotten, it's fallen off the tree. And when the maggots mature, they jump off the fruit, pupate, emerge as flies, and they retain the memory. They retain the memory of where they were born. And so when they want to find mates, they go to hawthorn on the fruit. Now, since we have apples growing in the same region that had been brought by colonists, one group of flies just accidentally didn't remember hawthorn very well and ended up on an apple. Okay, so and it only took just a pair of flies to do this. And this probably happened sometime in the 1850s or earlier. And so that led to a new population, a new race of flies that started breeding on apple. Okay, now they go to the fruit to mate. Okay, they're attracted to the fruit. So we have a population that is now going to hawthorn. That was the original population. Ones that are going to apple, and they don't mix with each other. And so even though both apple and hawthorn are in the same region, so it's sympatric, these two different groups, populations of Ragoletes, are diverging from each other. Now, the time when hawthorn fruits are out and apples are out, they do overlap, okay? They do overlap, but also the peak time when hawthorn fruits are available and apples, they're separated. Okay, let me just draw a picture. So if this is time going to the left, this is the availability of hawthorn. Okay, this time going that way through the year, and then apple, they're available something like that. Okay. Now, originally, of course, the peak population of the flies on hawthorn is going to be right here. Okay, the availability of flies pretty much follows the availability of the fruit. But so we had some of these late flies that were able to get on to apple, and now their peak abundance have shifted compared to the hawthorn flies. Furthermore, the ovipositor on the females, that's a device for laying eggs, has changed shape in the apple population compared to the hawthorn population. These are still regarded as the same species, but it is an example of sympatric speciation that appears to be in the process of happening. And among animals, I don't have too many other examples of this. Yes, Emily.
I was wondering why it isn't allopatric if it's like two different fruits. Isn't that like a geographical change?
Yeah. So with allopatric, they're in different geographical areas.
Mm-hmm.
Okay, the speciation, there's some sort of barrier that separates them geographically. Sympatric, they're in the same area.
Mm-hmm.
And that's where the speciation happens.
Oh, so it doesn't matter if they're on different fruits; they're in the same environment.
The same region. You know, both the plants are intermixed with each other in the same territory. So it isn't a separation of their ranges. It's sort of like it is a separation of their preferred microhabitat. Okay, because within a range, it isn't uniform. You know, there's areas that are a little bit, you know, different, and it's sort of related to patchy environment that I had mentioned earlier. Okay, does that get your question?
Yeah, thank you.
Okay. Okay, now even this is very rare. Sympatric plants, sympatric speciation does seem to be pretty common. It's not so common for animals, but this is another speciation mode called parapatric. And a lot of times it's hard to really differentiate this from allopatric because it's still a barrier. So here we get speciation with a very sharp environmental change. Okay, but they're adjacent to each other. And the only good example I can come up with is a grostis. This is a type of grass that lives in Wales. So there was a lot of mining activity in the past, and a lot of the soil that was dug out of the mines contains lead, and therefore there's this spread all over the place. Away from the mining part, and so the soil is contaminated with lead. And so let's have a mine right here, and all the soil that was dug out that is contaminated with lead spread on a region here. And initially, this was poisonous. Okay, it was too toxic for the grass. The agrostis species, it would grow out here, but not in this lead contaminated soil. However, eventually some plants started growing here. And of course, strong natural selection against the lead toxicity led to them diverging from the other populations of the grass that was living on normal soil. And now we have a totally new species. It's regarded as a new species because we can't cross the lead-free grass with the lead-tolerant grasses. They are now reproductively isolated from each other. And so this speciation event has taken place over the last 300 years or so. And so this is regarded as parapatric. There is a separation here, you know, but it's a very sharp boundary. With allopatric speciation, a lot of times the barrier is much, much wider. And so the two diverging populations are widely separated from each other. But here there's a real sharp boundary. And this is parapatric speciation. This is about the only example of parapatric speciation I've ever come across. Okay, any questions? Okay, so those are the modes of speciation. In all cases, allopatric, sympatric, and parapatric, we have to have some separation of gene pool. In allopatric, that is the easiest one to understand, and it seems to be the most common form of speciation, is that some physical barrier separates the gene pools. Sympatric, it's not a physical barrier, it's some sort of behavioral barrier or separation that leads to populations not mating with each other, and eventually they have to diverge. Okay, so what does this have to do? Speciation, it's a relationship to phylogeny and classification. Now, yesterday in lab, I sort of got into this. What do we mean by phylogeny? This is the result of okay, down through time, changes in the DNA, changes in the environment, natural selection, speciation. It leads to phylogenies. Okay, this is the evolutionary history of a group of organisms. And it's a lineage of ancestor, descendant, parent, offspring relationships where genes are being passed down through the generations and changing. So there's really two meanings to this phylogeny. It's the evolutionary history of a lineage, including the speciation events, but it's also a diagram, a family tree, an evolutionary tree that shows the genetic relationships between ancestors and descendants. And that's what we're going to be looking at on Monday in the lab. So this phylogeny that depicts cladogenesis, okay, the events of speciation. A lot of times these trees, they try to show genetic divergence, but most of the time the phylogenies are just showing cladogenesis, all right? So this term clade, I mentioned it yesterday, it's any branch of the evolutionary tree, okay? Just any branch, and all of the descendant species being derived from some common ancestor, and that common ancestor is represented as a node on the evolutionary tree, okay? Now, I mentioned yesterday, of course, the modern science, it's called, more often called systematics. It's a branch of biology that is concerned with figuring out the phylogenetic relationships of organisms and then classifying them. It used to be called taxonomy. You still come across that word quite a bit as well. Taxonomy is mostly emphasizing the groupings of organisms, putting them into species, genera, and other classification categories. Here, however, modern systematists, they first of all have to start out by trying to understand the phylogeny, right? Taxon, this is any group that can be classified is called a taxon. Today, we want a taxon to be monophyletic, okay? And I mentioned yesterday that in the past, due to Linnaean classifications, other types of paraphyletic groups were created. And so these are the definitions of a monophyletic group, and we can also refer to it as monophyly. It's any grouping of any taxon that we have that represents a clade of the evolutionary tree. And it must include the most recent common ancestor of that clade and all of its descendants. Okay, can't leave them out. Whereas a paraphyletic group is one that consists, it's a clade, still clade on the evolutionary tree, and it does include the most recent common ancestor of the members of that grouping, but it doesn't include all of them, and that is a paraphyletic group. These were often created by classification systems in the past. Since the 1980s or so, most systematists do not like them, okay? And so we insist on taxons in classification groups to be monophyletic. And there's still some paraphyletic groups that are out there in the classification because they haven't been worked on and haven't been figured out. What is never accepted in a classification scheme is polyphyly. Okay, this is a grouping of species together that it doesn't include the most recent common ancestor of the members of the group, nor does it include all of the descendants of that most recent common ancestor. And so these are three different terms. This is a figure in your textbook that is showing them. Okay, so in the circle here, this is a monophyletic group, okay, because it includes, right at this node, this is the most recent common ancestor, and all of the descendants are involved in here. Okay, yes, Adani.
What did you say that polyphyletic taxon's not accepted in?
Oh, well, no one would accept a polyphyletic taxon in classification.
It's just like went the correct way.
Yeah. They have been proposed in the past, but it's no one would never accept that. Back from the 1960s on to the 1980s, however, there were two groups of systematists. One of them were called cladist, the others called themselves evolutionary systematists, and they had some very, very heated debates about whether monophyletic or paraphyletic groups could be allowed. The cladist, they insisted on monophyly, whereas they called themselves evolutionary taxonomists. They said that a paraphyletic group is okay. You know, it makes sense that they diverge. If one group happens to diverge significantly, there's no problem with it. But they lost. Okay, so today monophyletic groups are the only ones that are allowed in the classification. Okay, and so this up here on the far right, this is a paraphyletic taxon. This is the most recent common ancestor of all the members that are the terminal branches, terminal ends of the branches. This means the species that are alive today, put together in the group, but it's excluding this part over here, which are also descendants of that most recent common ancestor. Now, this here is, I'm circling, that is also a monophyletic group. We could classify it separately or give it a taxon name. It's a more or less inclusive group, but this here is its most recent common ancestor, and it includes all of the descendants. And so we are striving for monophyletic groups. Okay, so how do you figure out the phylogeny? So the starting point of doing any sort of classification is to, first of all, figure out their evolutionary relationships, their phylogeny. And so you have to have evidence for that. And some of this evidence, of course, is fossils. Fossils are very important, but as I've already mentioned before, the fossil record is very incomplete, especially for terrestrial organisms. Some organisms have a pretty good, marine organisms can have a pretty good fossil record, but even here, the Earth has been dynamic. So first of all, we have a sea basin that would gather sediments year after year. The Earth changes. We get uplift, and that area of the sea that has its sedimentary rock becomes land. Now it's subject to erosion, and as it and several layers of sedimentary rock, along with the fossils on the top, can be completely eroded away, and then it may be submerged again, and we get new.
Did he just?
Is he glitching?
Not here.
Oh,
I think you froze.
You froze for a bit, Professor.
Oh,
Hello? Hello? Uh, hello, hello? Yeah,
I lost internet. Do you guys still have internet? I think the problem was on my side here.
yeah, we were just
No.
confused because you kind of froze and then left.
Yeah. My home network went completely offline, and so I had to wait for it to come back. All right, so we'll get back with this. Just a moment. Okay, it looks like a lot of stuff on my computer is all messed up. Okay. Okay. All right. So continuing, fossils are important. For one thing, they can tell us some characters of the past, but even what's preserved as a fossil, however, is selective. It isn't all of the organism. It's usually hard parts, and so like teeth, bone. Other types of information would be very important for constructing relationships among species. Would be, for example, flower color. That's never preserved in a fossil. And even with a lot of animals, their skin is not there because of soft parts, the muscles, and so forth. Rare cases that can be, but for the most part, it's bones, hard part. Some information might be for determining relationships between species is also whether they're diploid or they're tetraploid, or how many chromosomes do they have, and so forth. And none of that is preserved for the most part in fossils, unless it's something that has been died very recently within the last 50,000 years or so. We can't get to genetic information. So a lot of the information for phylogeny reconstruction comes from the living species that we have today. We're comparing them together. In fact, Cuvier, he started this with the comparative anatomy. So comparing the body parts between different species and looking for characters that indicate their relationship is very, very important. Now, with the advent of DNA sequencing, that has provided Systemdus with a treasure trove of information that can be used for phylogeny reconstruction. So we're looking for similarities. Organisms that have body parts or DNA that is very, very similar to each other must be closely related compared to larger differences in anatomy and larger differences in DNA sequences. However, similarity is not just not the whole game. We have to make use of the similarities in a logical sense. Not all the similarities that we find comparing two different species together may be useful for determining their phylogenetic relationship, because some of these similarities may be ancient, and therefore they arose far down the evolutionary tree. Some of these changes and similarities may be more recent, and therefore the more recent similarities are useful for telling who is more closely related. Okay, so we can look at these characters. Anything that we're trying to compare, we can call a character. And one thing is they have to be homologous. Okay, homologous characters. We cannot compare analogous characters. Analogous characters give us absolutely no indication of evolutionary relationship. Now, if you remember what homology is, I mentioned that some time ago. It is a similarity in a character that the reason for the similarity is because it was inherited from a common ancestor. And so only homologous characters give any sort of indication as to ancestry. Now, this homologous character, they may just only look the same or have some components in common. They may have even the same function, but they might have different functions. And I gave an example of homology before with the pterodactyl forelimb in tetrapods that has the same internal bone structure from elbow. And we could say this of the hind legs as well, okay, with a femur, tibia, fibula, and so forth. It's very similar, but still we're concentrating on the forelimb because these functionally sometimes are quite different, although the underlying bone structure is the same. And so like in humans, we use it to manipulate things, a cat to capture prey, also to walk on, a whale to swim, a bat to fly. So totally different functions. Now, when I'm saying homologous characters, it doesn't necessarily mean same underlying architecture, but different function. They could have the same function still, okay? But we're looking for that type of similarity. And so this is a good example of homology. All right, Now, even with homologous characters, okay, let me go back here because I just mentioned analogous characters. These are due to convergent evolution, not from inheritance from a common ancestor. They're not genealogically related. For example, wings of insects and wings of birds. Okay, they're both organs used for flying. So similar function in this case, but their underlying architecture is totally different. And therefore, that's convergent evolution. Another good example of convergent evolution would be the fins of fish and the fins of whales. Okay, they're streamlined, they're paddle-like, so that they can push water and swim, but that is convergent. Okay, there's certain qualities for swimming and living in water that are selected, okay, because it's similar selective forces that are choosing for the evolution of this particular form of limb, but it's convergent because the fins of the fish, they're architecturally different than what we have here with the whale fin. And so convergent evolution. So the only characters that can be used for phylogeny reconstruction are those that we know are genealogical, okay? That is homologous characters. But even here, we need to make a distinction between homologous characters. Okay, here's analogous characters. I forgot that I had this here, okay? Even though the bone, yeah, this is an interesting case of analogous characters. If we're asking about the wings in pterosaur, extinct, these incidentally, pterosaurs are not dinosaurs. Okay, I'm going to clear up your idea that your childhood gave you about what dinosaurs are. They're really more specific. So these are not flying dinosaurs, but the pterosaurs, they're extinct. They evolved flight. Birds evolved flight. And of course, bats evolved flight. If we're looking at the bone structure, that the bone structure is homologous, but the use of the forelimbs as flight devices, a device for flight, that is analogous. Flight was evolved separately in birds, in pterosaurs, and in bats. So the fact that they have wings and can fly, this is not a homologous character. It is convergent. So sometimes you have to really pay attention because a certain character, how you define it, might be convergent, or it might, at a different scale, be homologous. Okay, so of homologous characters, we can make two different distinctions. And this was important person who usually doesn't make it into textbooks. Why, I don't know, because he was the one who came up with these ideas. An entomologist from Germany named Willy Hennig, okay, back in the 1960s. He made the distinctions between homologous characters as to whether they are ancestral characters or recently derived characters. And he came up with these words, synapomorphies. That's a novelty in a character that has arisen more recently in the evolutionary tree. And therefore it gives a good indication of close recent speciation. However, some of these homologous characters that similarities that we look at may have occurred further down the evolutionary tree. And therefore these are ancestral characters. And therefore we call them symplesiomorphies. So synapomorphies, and very often I'll leave off the syn and the sim and just be calling these apomorphies plesiomorphies. So an apomorphy is a homologous character that arose recently, whereas a plesiomorphy is a homologous character that rose further down the evolutionary tree. And when we put the syn and the sim as prefixes on these words, it means shared, okay? So a synapomorphy is a shared derived character that's shared among two or more species, okay? Whereas a symplesiomorphy is a plesiomorphy, an ancestral homology, an ancestral character that is shared among a large number of different species or genera, okay? So a lot of times I don't use the syn, I just say apomorphy and plesiomorphy. Okay, all right, so an example here, if we can, so any homologous character needs to be regarded or determined as to whether it is a apomorphy, derived, or a plesiomorphy, an ancestral. So let's go back to the pinodactyl limb and let's look at a horse, zebra, a human, and a lizard, okay? All of them have the same bone structure in the forelimbs. However, if we look at the number of digits, the number of fingers that they have, how many fingers does a human have?
5.
How many?
5.
5. The lizard. The lizard.
5.
It should have 5 too.
5. What about a horse?
It has hooves.
It has hooves, but those hooves had to come from somewhere.
Yeah. It has like 2 of them, right?
What do you think that hoof, that horny material is? It's the same as your fingernails.
Oh, damn.
So how many fingers does it look like a horse has?
5.
Do you see 5 fingers on a hoof?
No.
It's 2, but I'm sure there's a bigger story to that.
I only see 1.
I mean, okay, yeah.
Okay, there's 1 hoof, 1 fingernail. Okay, so the thing is, horses and zebras, they have 1 digit. Okay, actually, they have 3 because on each side, but it doesn't protrude. On each side of that large finger, there are 2 bones on left and right. They're called splint bones. Those are the remnants of 2 fingers. In the embryo, the developing embryo starts with 5 digits. And these 2 outer ones, which corresponds to our thumb and our little pinky, they eventually disappear. And so in the leg, the other 2, ring finger and index finger, they become the splint bones, but they never are out as fingers. And so the horse is walking on the middle finger, okay? Think about what it's trying to say to you, all right? Bad joke, never mind. You may not get it anyway. All right, but so the horse and zebra, okay, still these came from the same ancestor, but as things evolve, you know, characters change. Now, if we want to look at this, which of these 2, we'll divide them into 2 different character states. It's the same character. The character is number of digits. The number of digits is the character. And what we have here among our collection is that 5 fingers, 5 digits, or 1 digit. Which one is more ancient and which one is more recent? Do you think? You think 5 fingers or 1? So we have 2 different character states. Which one do you think came first?
5 fingers.
5 fingers. All right, so that's the plesiomorphic state, having 5 fingers. Whereas having 1 reduced fingers down to 1, that is a recent change. It's a novelty, it's a derived character. And it holds together horses and zebras as closely related to each other, okay? Now, first of all, a lot of times when we do this type of stuff, how can we determine which one is a symplesiomorphy or synapomorphy? So we're taking a character, we have to define a character and determine character states. Okay, so in this case, the character is number of digits. And we have 2 character states, either 5 digits or 1. And therefore, one of them had to come earlier in the evolutionary tree, and another one is more recent. It's the synapomorphy that is really important for determining close genetic relationship. Okay, and that's what we're looking for, is the synapomorphies. So how can we tell the difference between a plesiomorphy or a synapomorphy? So I've already said this about character state. What we're doing in a phylogenetic analysis, which you will be doing on Monday, is that we have to divide our critters into an in group and an out group. Okay, so our in group are the taxa that we're trying to determine their evolutionary relationship. And so we know all these are related. So I have here species A, B, C, and D. We know that they're evolutionary related because they have some character that is unique to them. But we also have a bunch of other characters, and we see that there's 2 different or sometimes more character states among that homologous character. How can we determine which one is a plesiomorphy and which one is the apomorphy? Now, I'm going to be doing this, going through this with only 2 character states, but a lot of times we may have 3 or even more character states within our in group. So our in group is the group that we're trying to derive their evolutionary relationships from. Okay, and we know that they are a monophyletic group. We have some evidence of that by the use of some other types of characters, but we have all these character states, and I'm only going to stick with 2 character states per character. We need to polarize the character states. When I say polarizing character states, that means that we're trying to determine which of the character state came early and which one came late. Okay, in other words, which one is the plesiomorphy, which one is the apomorphy. Apomorphies are recently derived. Plesiomorphies are character states that are ancient, okay, ancestral. To do this, we have to compare it to a closer related group, another clade, which we refer to as the outgroup. Our analysis is not the outgroup. We're using the outgroup for what is called outgroup comparison to polarize our character states. So we're looking at another, and theoretically, the outgroup can be anything that is not in the in-group. And so if we have here a bunch of canines, okay, dogs, coyotes, and so forth, that we're trying to understand their phylogeny, bacteria could be an outgroup, but it doesn't make sense because they're so far away. Our in-group and out-group hardly have anything in common. And so what you're looking for is an out-group that's closely related to your in-group. All right, that's what we're going. Okay, now there's character states within the out-group. Okay, so we go back to our lizard and our human, and in fact, let's do that. Let me get rid of this. Our in-group, human, horse. Oh, those both begin with an H. Okay, this is our human, zebra, lizard. Okay. We know that this is a monophyletic group because all of these, the horse, zebra, the human, and the lizard, they have an amniotic egg, okay, which is unique. No other groups of organisms have amniotic eggs. And so they belong to what I'll be calling a little bit later amniota. All right, so we know that it's monophyletic. But we have 2 character states in terms of the number of fingers, number of digits. We either have 5 or we have 1, 2 different character states. So we have to appeal to an outgroup that is comparable. And a good outgroup would be amphibians, a frog. That's outside of the amniota. I mean, the frogs don't have an amnion. They don't have these extracellular or extra embryonic tissues such as the amnion and so forth. And therefore, it's a good outgroup. And so we can compare the number of digits that it has. And so actually, they have 4 digits, but in the embryo, they have 5. Okay, so let's say that they have 5 fingers. So within, well, we can, instead of frogs, let's get rid of frogs. Let's use a salamander. Salamander is even better. Okay, get rid of the frog, use a salamander. Okay, salamanders have five 5 digits. Okay, they have 5 digits. So we have 5 digits here. You have 5 digits. Whoops, it's the wrong place. 5 digits with the human, 5 digits with the lizard, 1 digit with the horse, 1 digit with the zebra. Now, since we have 5 digits in both our out group and in some members of our in group, 5 fingers, 5 digits must have arisen long ago. So a character state that's found in both the in group and out group must be ancient and must be an ancestral character. Whereas a character state that's only found within the in group, not all the in group, but some members of the in group must have arisen within that in group. Okay, therefore, the ancestors of horse and zebra have evolved that particular apomorphy. Okay, so our phylogeny right now, okay, I'm going to redraw it over here, is that we know that there's a branch here. This is our salamander, our outgroup, and human and lizard. We have, due to our evidence, we have an apomorphy here that now links our horse and zebra. Right at this, so the ancestor of the horse and zebra must have evolved a character state, an apomorphic character state, by the reduction of their fingers, okay? And so that's an apomorphy, and it's the apomorphy that gives evidence of the close relationship. Okay, so this is in principle how it works. Let's look at more than one character. Let's look at all of the vertebrates, okay? This particular group of organisms to which we belong. So this involves, what you do with a phylogenetic analysis is that we can't just use one character. The more characters we have, the better the evidence of being able to figure out the phylogeny. Now, this is a sort of simplistic data matrix. It's only plus and minus, and a lot of times data matrices will have other character states instead of just plus and minus, but this is what your textbook use. And so I'm going to use it as an example here. And so we're looking at all these vertebrates, lancets, lampreys, sharks, bony fish, amphibians, mammals, turtles, and lizards, crocodiles, and birds. And we have several different characters, each of which can be turned into 2 character states. Unfortunately, this data matrix is that their character states is presence or absence, okay? Not like my examples of 5 digits versus 1 digit, but still, we'll go through it. So we make a data matrix, and we then put the character state for vertebrae, okay? For example, vertebrae, bony backbone, okay? Down around the spinal cord. Lancets don't have a backbone, okay? And but all these other animals do, okay? So if we use the lancets as the outgroup, okay, we're going to use the lancets as the outgroup. They lack all these things. And so our in group is going to be from lampreys through birds, okay? So now this is sort of ambiguous because all of these with backbones inside, lampreys all the way to birds, they have a backbone, and our outgroup does not. Now that is ambiguous because the outgroup evolves too. And so what would have happened if lancets had a backbone but then lost it? Okay, so when we have this type of ambiguity, what we have to do is we draw on another outgroup that's further down the tree. Okay, a second alternative outgroup, which we can do with say a sea cucumber. These are echinoderms. They're related to starfish, and starfish or echinoderms and vertebrates are related to each other more distantly. Well, they don't have a backbone either. And so since we have both lancets as one outgroup and an alternate outgroup that both do not have backbones, then that makes this an apomorphy, the presence of a backbone. All right, so we can put that, separate off the lancets, and we put a tick here in an internode somewhere between the ancestor of lancets and the rest of the vertebrates that vertebrae had to evolve, right? So we go to the next one, jaws. Okay, now we can use our lancets as the outgroup. So we have lampreys that do not have jaws, but everyone else does. Okay, so here the plesiomorphy must be the lack of jaws, because we find that character state in both the outgroup and at least one member of the in-group. So it must have evolved, it must have been an ancestral condition. The jaws is shared amongst other members of our in-group, and it must have arisen right here, okay? Between the ancestor of the lampreys and the rest of the vertebrates. And we keep going. And so when we completely work out our phylogeny here, this is how we've no conflict. We have everything worked out. Now for feathers, that is unique to birds. And we can see that down here. Now notice here with the mammals, mammary glands, these produce milk. The outgroup doesn't have it, and most of the members of the in-group don't have it either. It's just the mammals. And therefore, that is a unique apomorphy for the mammals. It is derived. So this is how you put together and build a phylogeny. Now this can also use molecular characters. This has given us, as I said, a wide amount of characters, but our DNA sequences have to be similar enough that we can align them. Okay, similar enough that we can align them. Some parts of our genome have evolved so much that we don't see any comparisons, therefore we don't know whether they're homologous or not. But so we're looking at sequences that are homologous, and there's enough commonality there that we can align them. So how do we do this? Here's aligned sequences, and I have 12 different positions. So I have 12 different characters. Now, out of this 12 different characters, or within each character, we can have 4 different character states, the 4 different nucleotide bases. So our in-group here is species A, B, and C, just 3. We're trying to figure out, we know that they're monophyletic, but we don't know their relationships, and we're trying to build a phylogeny here. And we're using an outgroup, okay? So if we look at character 1, okay, character 1, what do you say as to what is the apomorphy, what is the plesiomorphy?
C is plesiomorphy and A is apomorphy.
Okay, all right, so this gives us some information, all right? So let's put a tree down here, and we have our A. I put the node as a tritomy, okay? It's unresolved. We have these 3 species, A, B, C, okay, and our outgroup. Okay, so since C is in both the outgroup and in some members of the in-group, whereas A is only within the in-group, it is an apomorphy for A. And I'm just going to put here 1' derived. But you see, it really didn't help us resolve any sort of relationships because it's unique to just only one species. And therefore, it isn't all that informative and doesn't help us all that much. It guarantees that all the members of species A happen to be, or yeah, all the species of species A or B. I put that in the wrong place. Sorry about that. Let me go back here and fix that. All right, I'll redraw it. A, B, C, and our outgroup. So this only happened in B. I'm going to put 1'. Okay, so that does give us indication that everyone in species B is monophyletic. Okay, it supports the monophyly of that, but it doesn't resolve our tree. So let's go to the next character. Character 2, what do you say about it? Two character states there. Which one is the apomorphy? Which one's the plesiomorphy?
G is apomorphies.
Okay, but the outgroups could be evolving too. It could be that G happened way down here, and on going to the outgroup that it changed to C. That would still explain then why A, B, and C all have G. In this particular case, it is ambiguous because we don't know, because in order to tell that it is ancestral, a plesiomorphy, it has to occur in both the outgroup and some members of the in-group. Here we have a case where we have one thing in the outgroup and something totally different in the in-group, and therefore we can't define that as an apomorphy or a plesiomorphy. What we'd have to do in this case is come up with another outgroup, an alternate outgroup further down the tree. That is related to our problem. And so when we have this type of ambiguity, what we're doing temporarily, we're putting the first outgroup as part of our in-group, just temporarily, and appealing to another outgroup, all right? And we'll come back to that and resolve that momentarily. Now, 3 is non-informative. It gives us no information whatsoever because everyone has the same thing. We go to 4. Now, what can you say here about 4?
A is more recent.
Okay, so T, since that occurs both within our in-group and in the out-group, that's the ancestral, the plesiomorphy, and A, and this here is now a syn apomorphy, because it's shared between A and B. And so if I can do this without destroying my phylogeny, get E. Oh, darn it. I got rid of it. Let me go somewhere else to draw this. Okay, now what we have with that evidence is that species A and B now we had on B 1 prime, but now we can say that they're joined together by an apomorphy, character 4. Here's our outgroup. Okay, so now we have a little bit of we've resolved a little bit of this. Okay, we see that there's one character here that is holding together a closer relationship that A and B have to each other than which either one of them have with C. But that's just only one bit of evidence. Let's go on. What's happened here? Okay. Go to character 5. What do you say about character 5? What is apomorphy?
C.
C. And what is it joining together?
A and B.
Joining together A and B. And therefore now we have even more evidence here. We have 2 characters that support a sister group relationship between A and B. Go back, going on down. I don't know. Do you guys see the screen turning green?
Yes.
Okay, I have no idea why that's doing that to me. Okay, we got to 5. 6. What do you say about 6?
C is apomorphy with the T.
T. Okay, and once again, it isn't very informative because it only affects species C, and therefore, but still, let's put it in there. 6 occurs here. Okay. Incidentally, when a syn apomorphy means it's a shared apomorphy, when it happens to be an apomorphy that's only in not shared and it's in one, it's called an out apomorphy. AUT, out apomorphy. And so it gives us evidence of the monophyly of species C. It supports that, but it's nothing that is shared with other groups, and it's only shared apomorphies that give us indication of relationships. All right, so we go on here, go to character 7. Yeah. What do you say about 7?
A is apomorphy.
Yeah, and it's only in species A. So 7 would be an out apomorphy. Has to occur here. And then what do you say about 8?
Is the same.
Yeah, so it's non-informative. And so we don't even use it because it's the same all the way across. What about 9?
G is apomorphy.
Okay, G is apomorphy and it only affects species B. So it had to occur here. Now, what about 10?
C is
Okay.
apomorphy.
Yep, C. And so that once again that's only species C. And let's go to 11. Non-informative. So we don't even put it on the tree because it doesn't tell us anything. And what about 12?
Ambiguous.
It's ambiguous. Okay, so we had 2 ambiguous, this one here, and where was the other one? It was
Number 2.
number 2, this one. Okay, so since these are ambiguous, what we do is we appeal to another outgroup. So an alternative outgroup. And so in this particular place, we're putting the outgroup, the original outgroup, in our in-group just for that one character, just for that one character. So if we're appealing to a second outgroup, what now looks like the apomorphy? The plesiomorphy has to occur in some members of our in-group and also in our outgroup. So we're jumping down the tree one more outgroup and treating the original outgroup as part of our in-group, just for this one character. That's all, to resolve the ambiguity. So
C.
2 of these are plesiomorphy, which is apomorphy.
C is plesiomorphy, and then G is apomorphy.
The G is an apomorphy, and the G is an apomorphy that arose right here. Which gives us now separation. Okay, I'll put the alt out here. Now notice that what I'm putting on the tree is not the plesiomorphies. You're only putting the apomorphies. Only the apomorphies. You never put the plesiomorphies on the tree. Okay, all right, so now we can go over here to this one, 12. And now what do you say about it? Because once again, we have to appeal to an outgroup because it was second outgroup, alternative outgroup because it was ambiguous. So looking at it now, what is the apomorphy?
A is apomorphy.
Apomorphy is A. And that occurs because don't want to give you the impression, okay, what character, that was 12, that the outgroup always has plesiomorphic characters. That's not the case. Now, I'm mostly giving examples here where we have all the apomorphies are within our in-group, but still the outgroups evolve too, okay? And I gave sort of an example inadvertently of a frog because I was trying to use it as an outgroup. I changed to salamander because it has 4 digits. It doesn't have 5. But we know that it lost 1 digit, okay, compared to the salamander. So even outgroups evolve, all right? And so the apomorphy has happened here in our outgroup. It would be incorrect to put it anywhere else, all right? So let's do more here. I'm going to add one more character, 13. What do you say about 13?
T are apomorphy.
Yeah, T is the apomorphy. But look what it's doing here. It's putting together B and C. It's putting together B and C, which we can't do here. So that particular character is supporting a closer relationship between B and C instead of A and B. So which, in other words, we have character conflict. And so in databases, we do have character conflict, but which one of these two solutions, A and B being sister groups or B and C being sister groups, which one do we accept? I only have 1 more minute, and so I'll say this real quickly. In cases like this, we have a principle called parsimony, and I'll go over this again on Tuesday. You choose whichever of the 2 relationships has the most support. Okay, so right now we have 2 characters that support an A and B relationship, and thus far only 1 character that supports B and C. And therefore, the more parsimonious outcome is that requires few evolutionary changes, as I'll explain on Tuesday, is accepting A and B as being closely related to each other by evolutionary scent. Okay, and so we call this, let me get up here and this will be the end, when we have a character that is in conflict, and this would be due to convergent evolution, okay? Convergent evolution, it is, just a minute here, I had the word on the tip of my tongue. I'll have to look it up. I had there's a special term for this. Oh yeah. Never mind. I'll say it on Tuesday. I'm going to have to look it up. I forgot what the term is because I don't come across it very often. All right, so this is where I'm going to stop, and this is also going to be the area for the quiz on Monday. Okay, because it's going to carry 3 lectures, 2 or Thursday of last week, Tuesday, and today. So where did we stop on Thursday? Or where did we start on Thursday last week? I think it was lecture 39 or lecture 1, slide 39. Is that what you have?
I think we stopped at 38.
38, okay. So from 39, lecture 1, 39, up to this slide 36 on lecture 2 is material for the quiz on Monday, lecture quiz. After that, it's going to be just only 2. Okay, any questions? Any slides you want to see?
Just for the last one, so do we